So now we're going to talk a little bit more about some of the co-cultivation applications and hopefully get some nice interesting pictures. So here we talk about asymmetric cell co-cultivation. So in this particular case, we have endothelial cells on the insides of the fiber. This is a different fiber that we work with. And here we can see vascular smooth muscle on the outside of the fiber, so you can see the three-dimensional growth characteristics of the cells on the fiber. In this particular case, we could grow endothelial cells on the inside of the fiber and subject them to chronic shear stress, defined levels of chronic shear stress as they are growing in vivo in the body. And shear stress is quite important to endothelial cells, and they respond to this shear stress in many different ways. But most specifically, they lay down flat, they form a monolayer, they form tight junctions, there's physiologic expression of Pilate bodies, and they stop dividing, most importantly. So I look at the endothelial cells. I don't look at endothelial cells. I look at the endothelium as an organ that's inside your body. You have about 1 kilogram worth of endothelial cells that line all the blood vessels. The surface area is frankly unimaginable. I see many different levels of calculation of surface area, and I'm really not sure which one is right, but a tremendous amount of surface area. And endothelial cells provide very important functions. They control the composition of the interstitial fluid, which cells are actually growing in, which is actually quite different than the composition of serum itself. They're responsible for the blood-brain barrier. They're responsible for cancer metastasis. They're responsible for stem cell homing. So the only way to really get true physiologically relevant endothelial cells is to grow them under shear. In this particular case, this is showing the endothelial cells flattened on the insides of the fiber. It's really remarkable to me just how flat they are and how thin they are. It's amazingly thin. It's not a complete monolayer in this particular case, but what we're going to be doing here is looking at cell transmigration through the fiber. This is Dr. Pepper and Elizabeth Walsby. They were in Wales, and now she's moved on. I forget exactly where, but she's still working on the system. And what they've done is set up a model for cancer metastasis. And so they find that they put in the endothelial cells and subject them to shear, and then they put a chemotactic agent on the ECS side of the fiber to attract the cancer cells, the circulating leukemic lymphocytes. Lo and behold, they wind up showing up on the outside of the fiber. So they specifically interact with the endothelial cells in the presence of shear and are able to, and I don't know how they do this, they're able to transmigrate through a 0.1 micron tortuous path, pore size, 200 micron wall thickness, and show up on the other side. And the two things that are important about this particular model is, number one, it's a way to get enough endothelial cells and enough of the circulating leukemic lymphocytes so that you can do genetic analysis, you can look at protein expression, you can look at receptor activation with enough cells here. It's about 80 square centimeters of surface area. And secondly, this process is quantifiable. So they can put in drug X and say, okay, we saw 80% reduction in cancer metastasis utilizing this drug. We also have a co-cultivation where we put bone marrow stroma onto the fibers, and then we can put hematopoietic stem cells on the outside of the fiber. And this is Dr. Mayasari Lim, who did some of this work. Very interesting stuff. And she found that hematopoietic stem cells cultured in this environment and grafted more rapidly when injected into skid mice. Another application that is I find very interesting and I'm happy to have been involved with. Whenever we're working with hollow fiber, we really want to look at recapitulating the in vivo environment as closely as possible. So this is Dr. Nigel Yarlett, Pace University, and we work with some folks at Tufts Veterinary School. And Cryptosporidium is a unicellular parasite that has never been cultured in vitro for all of its life cycles before. So it's a parasite of the gut epithelium. 40% of the kids in Africa are infected with Cryptosporidium. It's a wasting disease. It's kind of like cholera. It's not very pleasant at all. So we look at this and we say, okay, it's interesting to think that actually the gut is a low oxygen environment. So what they were able to do is take gastric epithelial cells and put them on the outside of the fiber, and of course the interior of the fiber is going to be a high oxygen environment, and then they devised a way to put oxygen scavengers into the extracapillary space of the fiber and generate a low oxygen side, which is going to be mimicking the oxygen intention that you find in the gut. And it took a special lipid package and a few other things, but they were actually able to take Cryptosporidium out of an animal, have it go through all its stages of development inside the cartridge, and then reinfect an animal. They'd never been able to do that before. In fact, I was at the CDC just yesterday, and there was a guy who had some tremendous experience with working with Cryptosporidium, and he still has to use cows as his vector, as his host to culture Cryptosporidium. And of course, it's very difficult to develop drugs against a particular disease if you don't have a way of growing it in the lab. So this is something that I'm very proud of, and I think is actually quite significant. Exosomes are something that's been kind of coming along just recently. It's only a few years ago. People thought that exosomes were just a way for cells to get rid of their intercellular garbage. They thought they were just garbage balls that cells would use to get rid of stuff they want to get rid of. The flow cytometry people would always say, down at the bottom of the flow plots, there'd be all these little dots of exosomes. It's like, I wish these things weren't here. What a pain to have them around. Now it seems that exosomes are involved in many, many different types of processes, specifically cellular regeneration, tissue regeneration. The environment of the circulating blood is designed to be very hostile to free RNA. We don't want to have free RNA floating around in the blood vessels, in the circulatory system, affecting cells downstream unless there's a specific purpose. And exosomes are targeted vesicles about 80 to 120 nanometers in size, and they're quite complex in terms of the proteins in the membrane. I believe their primary function is to protect microRNA and ensure that it gets delivered to specific sites in tissues downstream. So it's this ability to enhance tissue regeneration downstream that gives them very great possibilities for different types of therapeutic applications. So exosomes are secreted by cells, and normally the standard protocol for producing them is to take a flask and grow the cells up to confluence, and then remove the serum and grow them in serum-depleted media, serum starvation conditions, because serum contains endogenous exosomes that we don't want to have to, we can't purify away from. So this is not a very physiologic process, and this is some of our early data where we compared exosomes produced from human adipose-derived adult mesenchymal stem cells. So basically we can see that one C2011 bioreactor over a period of 6 weeks of harvesting can yield as many exosomes as about 700 T225 flasks, but at a volume of only 120 mils. Based upon this methodology, I calculate that we can actually easily produce gram quantities of exosomes utilizing 4 to 6 of our larger cartridges. And we can do this without contamination from serum endogenous exosomes. This is just an example of some of the things that we've done with exosomes. Of course, there's lots of other applications for exosomes. I believe we're going to see that exosomes are the prime therapeutic vector from stem cells, that the stem cells themselves are not necessarily what's producing the tissue regeneration, but the exosomes secreted by the stem cells. So this is a poor little rat that we've got a standard model for wound healing, and you can see under the red circles, that's where we've applied the exosomes, and it's really not very many of them. It's around 2.5 times 10 to the 6 exosomes applied to each wound versus the controls taking a lot longer to heal up. We have collaborated with a company in North Carolina to develop some of this data, ZenBio, and the president down there, he has a sister who happens to work in the cosmetics industry. And they produced so many exosomes with this cartridge. They had 25 milligrams of the stuff, and they didn't want to just throw it away. So he called her up and he said, "Hey sis, I got this stuff that seems to stimulate collagen regeneration and tissue healing and things like that. Any ideas what to do with it?" And she said, "Sure, let's put it into a face cream." So there's an actual face cream on the market. It's been fully tested and validated. You can order it online. This is not necessarily a commercial for it. It's called Exoface. And it contains 150 million exosomes per bottle. I like the term they use. It contains smarticles, and they call it Zen cubed technology because it was the third cartridge that they used. The maximum number of cells, they loaded 10 of the 9 cells into the cartridge, and that's how they got all these exosomes. So I believe this is the first real commercially successful application for exosomes. And I've given out a lot of samples of it to some of my female friends, and they swear by it. They think it's great stuff. So the advantages in hollow fiber for exosome production is that a large number of cells can be cultured in a small space. The treated exosomes are concentrated, so we don't have large volumes of supernatant that we have to deal with. We can do continuous production over several months. We're only working with a 293 or something like that. Serum can be used without contamination from endogenous exosomes, but I have to say at this point in time, I've been kind of surprised by the fact that we've yet to really find a cell type that we cannot adapt to CDMHD. Now, I'll be honest with you, we have not yet tried CDMHD with mesenchymal stem cells. That's still a work in progress. But CDMHD has been used successfully with BHK, CHO, 293, hybridomas, primary cancer cells, and cardiac spheroids. If we can get the cells up to high density, we seem to be able to adapt them over to CDMHD. And CDMHD is going to be validated for cGMP production of exosomes. Cell proliferation may be limited specifically with mesenchymal stem cells, and I think they're a separate issue, and we're going to talk about that in just a few slides. And this is just a little reminder of the plastic waste that's generated by 1 times 10 to the 9th cells, generating them in standard culture methods versus our cartridge. Another application that I'm just going to mention, it's quite significant for us because it allows direct mimicking of human bioavailability of antibiotics and modeling pharmacokinetics and pharmacodynamics. And the basic idea is that we can strain and retain the bacteria inside the cartridge, and then we have a circulating loop through the central reservoir, and then we can add and remove drug from the central reservoir, and the idea being that concentration of drug in the central reservoir equals concentration of drug on the bacteria, so that we can look at dosing profiles that result in optimum killing. We can look at dosing profiles that result in resistance. We can easily do 2 and 3 drug models, which is going to be, I think, kind of the wave of the future for the immediate future for antibiotic therapy as resistance is on the rise. It's also a good way that we can do time course dependent treatment of cells, lots of cells as well. For example, one of the things that we do with mesenchymal stem cells and exosomes is we want to look at heat shock. So we can just take the central reservoir and put it into a 45-degree water bath and try doing that with 200 flasks at one time. So this is also a way of doing time course exposure of various treatments to cells in a very defined and physiologically relevant fashion. Also, just another little reminder that FiberCell is a enabling supplier to NASA. But you have to think about how do you do cell culture in 0G. Everything has to be retained inside a sealed system. You can't pipette in space. You can't do cell culture in space. So we are an enabling supplier to NASA. So whenever you read about any cell culture experiments coming off the International Space Station, that'll be our cartridges that they're using for that. This is my final example that I like to show for just how profoundly cell culture conditions can affect cell physiology and that they need to be as in vivo-like as possible. This is a very interesting piece of data that we got from some collaborators, Thailand in fact, but this is culturing of primary cholangiocarcinoma cells in the hollow fiber bioreactor utilizing CDMHD versus the cells being grown in flask culture. And what I find interesting about this is, so what they're doing is they're looking to define a biomarker for cancer that they can use, this specific cancer that they can use for diagnostics of cholangiocarcinoma. So what we see in the B is the main piece of data we want to look at. So they utilized our 5 kD molecular weight cutoff cartridge and concentrated and collected the secretome of the cells, and they utilized CDMHD to culture the cells. So any protein present here in the ECS is going to be secreted from the cells. And they also defined the low amount of apoptosis. So these are really a reflection of the secreted products, not without a lot of intracellular protein contamination. So if we were just simply concentrating the secretome from these cells, then all the proteins along should be concentrated to the same degree. But in point of fact, the one protein NGAL is 56 times more secreted in hollow fiber in three-dimensional culture than in flask culture. So it's this cell culture environment is causing these cells to behave more physiologically, because then they went back and started looking at serum concentrations, different proteins, and found that in point of fact, NGAL was extremely elevated in circulating plasma of the patients, and they wouldn't have picked this up if they were doing this in flask culture. So just one example, another example of how profoundly cell culture conditions can affect the behavior and physiology of the cells.